Now here you see we will be dealing with the processes of recombinant DNA technology. Now, how we prepare a recombinant DNA, we have earlier we have done that. Now, in today's class, we'll be dealing with the processes of the recombinant DNA technology. Now, how does the industry go with it? Now, first and foremost, you see here we deal with the isolation of DNA. Once the DNA is isolated, the fragmentation of DNA, we use the restriction endonuclease, that is the there. And in the second, the isolation of the desired fragment. After you mark it, cut that, then you need to isolate the fragment. That means you take out the fragment from the medium. Now once you take that fragment medium, the ligation of the desired fragment in the vector, that means this recombinant DNA has to be prepared. That means after cutting, it has to be added with the vector DNA, and then after that vector is prepared, the transferring the recombinant into the host, that we have already done. For example, that cell bombarding the cell, that is gene jet, like that. Okay, then ultimately gene gun also. Then after that host cell, after transferring of that DNA, that host cell has to be cultured. Okay, now you see culturing of the host cell in a medium, and these are all done in a large scale. And then large scale extraction, for example, you have coded for the production of a particular enzyme, for example insulin. Okay, so artificial insulin that is produced through the bacteria Escherichia coli has been coded for that. So when large scale extraction of this desired product can be taken out. Okay, so these are the points that we would be dealing. Now firstly, when we would be dealing with the isolation of DNA, now what we all know that DNA is within the cell nucleus. Okay, so the main purpose how to take out the DNA is you have to break the cell wall to release the DNA. Okay, so now when the DNA is released, that histone molecules are also there. That DNA is basically it is negatively charged. So histone, with that histone, protein, RNA, many polysaccharides and lipids, whatever is there in the packaging material of the DNA has to be separated first. So that procedure is known as the purification of DNA. So while we are dealing with the purification of DNA can be isolated from plant cell, DNA can be isolated from the bacterial cell, DNA can be isolated from the fungal cell. So here you see these are certain points. For example, enzymes treatment is done to release them. For example, lysozyme. So lysozyme is used when the DNA, I have written the short form, you see it is actually the bacterial cell, lysozyme. Then we have the cellulase. Cellulase is used when the DNA is extracted from the plant cell, and chitinase, here you see chitinase is used when it is released from the fungal cell. Okay. Now when these things are isolated, the DNA has to be precipitated in chill ethanol. So when it is precipitated means sedimentation, like for formation of the DNA is there. So you have to do it in chilled ethanol, and when it is done in chilled ethanol, you can see take out very fine thread like fibers of DNA that has to be there. Now you see cutting of DNA, how it is cut. Basically how it is cut, the DNA is used with endonuclease, then again it is used by electrophoresis, where gel electrophoresis, where it is separated, and we use of the restriction endonuclease that is there. So these are the basic process. Now after that, once you get the gene, a very important thing is amplification of gene. Now how does amplification means? Multiplying this gene. Amplification means producing few million copies of that particular gene that has to be coded. Now they use polymerase chain reaction, that is PCR, that means multiplies copy of DNA produced in vitro. In vitro means within the cultured medium. What happens, the sequence, the base sequence like molecule is prepared and produced in the medium. So when that medium is produced, now they the DNA, they get ligase. That means they get attached to it, and these chemicals that is used, okay, that endonuclease, it helps into multiply that. Okay, now see this portion I'm coming. Now here you see this one, when DNA, it is precipitated, this in chilled ethanol, here you can see fine DNA threads can be taken out. Now next you see, this is the gene amplification by the PCR technique. Now see, this is the DNA, and this DNA, it cannot be, it is synthesized in vitro, as I told you. You see two sets of primers chemically synthesized. What is chemic primers? They are known as oligonucleotide. That means a pseudo thing that mimics the original DNA sequence. Now they are complementary to the region of DNA. You see here. Okay, and they use of DNA polymerase. The enzyme extends the primer using the nucleotide provided in the reaction and the genomic DNA template. See here, this is the primer. Okay, and here this is the original DNA. These two formations same. So this has to be multiplied. This portion has to be multiplied. So the use is DNA polymerization. This makes a copy. This also makes a copy. So when they are making a copy, you get an amplified billion times. See, only two base pairs, for examples, were there. Complementary pairs were here, and again they are made copies. And this, see, first each cycle has three steps. First one is denaturing, then the primer annihilation, and extension premier. See, this primer, they extend, this extends this side, this extends this side. Okay, so ultimately you have got a billion. See how many copies. Here you see such repeated amplification is achieved by the use of thermostable DNA. That means it is not denatured by heat, and this amplified filament, if desired now, can be used to ligate with a vector. Ligate means used. Now then, insertion of recombinant into the host, this we have done with the help of a vector. We have learnt about the various methods. One is ampicillin, antibiotic resistance with ampicillin, and another one was tetracycline. Then ultimately you see here, obtaining the foreign host product. Now when it is inserted, this recombinant DNA, when it is inserted in the host cell, now our motive is, after the clone, you see, after having the clone gene of interest and having optimized the condition to induce the expression of a target protein, that means the finished product. For example, if we are producing insulin, the production of insulin in a large scale, so that production, if any protein encoding gene expression in a heterologous host, we call it as a recombinant protein. So the cell harboring the cloned gene of interest may be grown in small scale in laboratories. Okay, the culture can be used to extract desired protein and then purifying them by using different separation technique. So for purification technique, here you see these, this one and this one. Okay, now these are stir tank bioreactors. When they are used over here, these are known as bioreactors. See, what is the definition? See, small volume of culture cannot yield, so we need to produce in large quantity, and they are done by bioreactors. Okay, now bioreactors, what do they use? See, for as raw materials, they use the DNA biologically converted into specific products. They use certain enzymes and optimum pH. See, what do they produces a growth medium and optimum growth conditions. That means the temperature, pH, substrate, salt, vitamin, minerals, whatever is there. And these stir tanks is, they keep on stirring the thing, and sometimes their gases are bubbled into it. Okay, so for the oxygen are bubbled, so that increases the reaction. Okay, now after completion, here you see the biostream processing. Now what is biostream processing? Now after completion of the biosynthetic stage, the product has to be subjected to series of processes. That means it has to be cleaned and purified and packed. separated from the other substances, and these products can be formulated for suitable preservatives and can be marketed. And they have got a strict quality control checking through it, and the products are required. The downstream processing are related to them. So what are basically downstream processing? They are related to the process which quality controlling tests of the various product. So after that you achieve, and after that you achieve the finished product. For example, here if insulin is produced, the insulin has to be extracted, purified, bottled, quality checked, and then marketed. So that after processes, that is the purified, bottled, checked, and marketed, comes under the downstream processing. Okay.